Is cellular respiration making your head explode? Actually, the cellular respiration that is going on inside your cells is keeping your cells going by making a bunch of ATP. But the last lecture may have made your head spin at least, and that's totally okay. That was the first time that I took you through that whole process, and we're going to go through it again, and we're going to take it a little bit farther. So let's review a bit, and let's switch to a different color. What was I using? I don't know. We'll go with this right here. C6 H12, because I've been using yellow for hydrogen. Let's stick with this. C6 H12O6 plus 6O2 yields 6 CO2 plus, dun dun dun, 6H2, for consistency's sake, O. There we go. This is the overall chemical reaction for aerobic cellular respiration. Oh, and FYI, the word aerobic means with oxygen, with O2. There you go. So that's why it's called aerobic cellular respiration. Now you already know that this chemical reaction doesn't happen at once. It's actually a summary of a whole bunch of different chemical reactions that I already took you through, like glycolysis, the intermediate step or transition step, it's called sometimes, and the citric acid cycle. And through glycolysis and the citric acid cycle, I'm going to erase that. All of the carbon in glucose gets converted to CO2. We even counted them up. We lost a couple during the transition step, and we lost all the rest, 4 more, in the citric acid cycle. So remember, this is an oxidation. It happens first. It happens first in glycolysis, the intermediate step, and the citric acid cycle. Put that in parentheses right there. And that's the part that I took you through. But what happened to these hydrogens here, you may ask? We already talked about this, that these hydrogens get picked up by NAD plus and NADH. Oh, let's get a different color for them. NAD plus and FAD, and they become NADH and FA. So it's the wrong color. Oh, can't use the wrong color. Let's go back and change that. What the heck? I think I used that. And FADH2. Remember? Get a whole bunch of these. So we ended up with a whole bunch of NADHs and just a handful of these FADH2s. Now, what did these guys do with those hydrogens that they pulled out of glucose? Think about that for a second. They removed these hydrogens from glucose and carted them to the mitochondria. Remember that? They went into the mitochondria. I need a different color for the mito. I think we used tangerine orange for that, right? So they carted them, they took those hydrogen atoms from glucose, took them into the mitochondria, where those hydrogens are used to make a whole bunch of ATP. NAD plus and FAD remove hydrogens from glucose. They were right in there. And they bring those hydrogen atoms to the electron transport system. Oops. Back to this. Where they're used to make a bunch of ATP. Now let's talk about how that happens. To do that, I need to draw a bigger version you know, just like this part here of the mitochondria. So let me do that. I need my mitochondria color here. And we'll go with that orange. It's a little closer. Okay, so here's the outer membrane of the mitochondria. Here's the inner membrane, right in there. Okay, back in the mitochondria, NADH and FADH2 are like, okay, now we're in the mitochondria. We're going to make a bunch of energy here. Let's, what color did I use? Oh, who cares? For the proteins of the electron transport system. These purple things here are proteins of the electron transport system, or the ETS. They're protein complexes. And I got this one over here that I draw to kind of look like a tall pilgrim hat. And I did it that way on purpose. There we go. Now, remember that these hydrogens came from glucose. C6H12O6. Here, let me even color code like that. Remember, NAD plus and FAD took these hydrogens little by little, one by one, out of glucose, and they bring them into the mitochondria. Where, once in the mitochondria, NADH is like, here, take your hydrogen atom back. But it doesn't give the hydrogen in the form of a hydrogen atom. It gives it up in the form of electrons and protons. So actually, if we look at this right here, let's just focus on FADH2 for a second here. FAD took on 2 hydrogen atoms in the form of 2 electrons and 2 protons. And remember that an electron plus a proton equals a hydrogen atom. They usually stay together, but in this case, they don't. The electrons get passed from protein to protein, and they're just kind of kept busy for a while, whereas the protons get pumped across the inner membrane. Right? NAD will do the same thing, NADH. Only NADH, here we go, NADH, I'll draw the H first because I'm already on yellow. NADH goes back to being NAD plus, and then it gives up this hydrogen atom, but 2 electrons actually go with it in this case, and 1 hydrogen ion, which is a proton. Remember, this thing is called a proton. This is just all review. This is all stuff we saw last time. All right, but remember, way back here when we added all this stuff up, hoping right here, how much NADH and FADH2 we got out of that? We got 10, remember? 10 NADHs out of one molecule of glucose. So back here, there are going to be 10 of these NADHs, so there are going to be 10 protons pumped across that inner membrane of the mitochondria. 1, 2, 3, 4, 5, 6, 7, 8, 9. I'm running out of room here. 10. Holy crap. You got a concentration of 10 protons in this intermembrane space and a concentration of zilch inside the mitochondria. That is called, friends, a concentration gradient, and you know that's not stable. A concentration gradient is not stable. So what's got to happen is that these protons need to come back in here to equalize it. Nature doesn't want 10 and 0. Nature wants 5 and 5. So 5 of these got to move through, but they don't diffuse back through just anywhere. They diffuse back through this one protein that looks like a pilgrim hat that is called, oops, whoa, what happened there? Accidentally hit that with my thumb. That is called ATP synthase. Get that out of the way. There we go. ATP synthase. I'm going to erase this stuff over here to give us a little more room. And here's the deal. Remember that when, take care of this too, when a gradient is equalized, okay, this situation that we've got here is very unstable. So when the gradient is equalized, energy is released. Let's write all this stuff down. Let's get a little sentence, a little paragraph going here. Okay. Let's say during glycolysis, NAD plus, and during glycolysis and the citric acid cycle, etc., NAD plus and FAD remove hydrogen atoms from glucose. They bring, let's say as NADH and FADH2, they bring the hydrogen atoms to the electron transport system in the mitochondria. I'm just going to write mito there. Okay, we got that. NADH and FADH2. We're still color coding here. I'm working hard to color code because I think it makes it a little bit easier for many of us. Okay, NADH and FADH2. Oops. We'll just keep it in gold for a little bit. Give up the hydrogen atoms to the ETS in the form of electrons and protons. All right, so we got that. The electrons get passed from protein to protein. While the protons, these protons here, get pumped across the inner membrane of the mitochondria. They get pumped. They get pumped across the inner membrane of the mito. And they get stuck in this intermembrane space here. This creates a concentration gradient that's not stable. Let's go back. Oh, let's pick a different color. This creates... Oh, man, I'm doing it again. This is not my first rodeo. I really need to turn there. There we go. Apologies. Good grief. I can't believe I did this. This makes it a lot easier for you to see. My bad. I guess I was just so excited I wanted you to be able to see this up here while I was writing all this down here, but, well, you can't have everything, I suppose. But I'd rather be able to see at this point. All right, so this creates a concentration gradient. Concentration gradient of protons. That's not stable. There we go. Okay, we'll come right back to this. I just want to show you the picture again. All right. So they osmose back into the mitochondria, right? They're osmosing, which is a type of diffusion. It's diffusion across the semipermeable membrane. But they release energy when they equalize that gradient. You remember that sentence that I had you circle and put stars and swirlies all around? Energy is released when these protons flow back through this enzyme ATP synthase. And it's kind of like, remember those little water wheels that you played with that little kids play with at a pool or a beach, and it's got like this paddle wheel on it, and you pour sand or water over it, and the water makes the wheel go around? The flow of these protons is like pouring sand over a water wheel. ATP synthase is like a water wheel. And it physically gets turned around as those protons flow through it. This is mechanical energy. This is what I'm talking about, how energy is released when this gradient is equalized by having those protons flow through it. It is like water falling over a dam and making a water wheel turn. That is mechanical energy. And incidentally, that's how the hydroelectric power works. That's why we build dams. That's why there's a dam in Holyoke. We have hydroelectric power, and the flow of the water over the dam basically turns a wheel that's attached to a generator. ATP synthase is like that wheel, all right? And it uses that energy. It's being able to physically turn because of those protons to take ADP and put that phosphate group back onto it to form ATP. So instead of electrical power here, this is mechanical power used to put ADP back together with its initial phosphate group to make ATP. So freaking cool. It is like water falling over a dam. Go back and listen to that again, because I really want it to sink in. That these protons setting up a concentration gradient that's not stable is like water being held back by a dam, and when they equalize it through ATP synthase right here, it physically turns that enzyme, like turning a water wheel on a generator, and that gives ATP synthase the energy to put that phosphate group back onto ADP to make ATP. Your cells use the hydrogen atoms that come from the carbs you eat to make ATP. It is freaking fascinating. It is so cool. Like if you look back up here, and if you look at C6H12O6 right here, your cells aren't like, ooh, carbon atoms, got to get me some of that, love that. No, your cells just spit all those carbon atoms out as CO2. What gets your cells excited are like, oh my God, hydrogen atoms, and there are 12 of them in here. That is so awesome. We are going to rip those out, and NAD plus and FADH are going to rip those out of glucose and the carbs you eat, cart them into the mitochondria, where they're going to, NADH, going to give up those hydrogens in the form of 2 electrons and protons. Those protons are going to get pumped across the inner membrane of the mitochondria, set up this absolutely awesome concentration gradient that's like water being held back by a dam. And as the protons equalize the gradient by flowing through ATP synthase, that gives ATP synthase the energy that it needs to put this phosphate group back onto ADP to make ATP. So freaking cool. Your cells make energy through diffusion, and they use protons that come from hydrogen atoms to do it. Back on our very first remote lecture that I updated for you, I talked about transport across the membrane and diffusion, and you've all done a virtual lab by now on diffusion and osmosis. And I mean, I'll confess to you right now that when it's time for me to teach diffusion, I'm like, oh my God, kill me now. It is so boring. I'm like, oh, diffusion. It's like putting a drop of, blue food coloring into a glass of water, and you've got a high concentration of dye molecules here and a low concentration out here, and those molecules all want to spread out. They diffuse. I'm like, oh my God, this is so boring. But it's not boring. It's really, really, really exciting. Diffusion is how your cells make energy. It is one of the most important concepts to the cell. It's one of the most important things you can learn about how your cells work, because diffusion across that semi-permeable membrane is how your cells make ATP that your cells need all the time. So let's catch up with our excellent paragraph here. Let's review this again. During glycolysis, NAD and FAD remove hydrogen atoms from glucose. They bring those hydrogen atoms, they become NADH and FADH2, and they bring those hydrogen atoms to the electron transport system in the mitochondria. Once they're in the mitochondria, NADH and FADH2 give up the hydrogen atoms to the ETS in the form of electrons and protons. They're splitting up for the first time. Those electrons get passed from protein to protein, while the protons, that should be plural, get pumped across the inner membrane of the mitochondria. This creates a concentration gradient of protons that is not stable. And here's a super important sentence, so we need a special color for that. What's a color we haven't used that's really awesome? How about orange? All right. Energy is released when the protons equalize the gradient through ATP, oops, need a little more room here, through ATP synthase. ATP synthase uses this energy to take that initial phosphate group, add it back onto ADP to form ATP. And that's an endergonic reaction. It requires the input of energy to do that. Okay. This whole process is called chemiosmosis. Called chemiosmosis. Alrighty. Now I want to talk a little bit about how much ATP do we get from this process. Do we just get one? No. Like how many times does ATP synthase turn when protons flow through them? So as a rule, or rather, let's say under ideal conditions, which, hardly ever happens, but under ideal conditions, for every NADH, you get 3 ATP. And for every FAD, I know this isn't going to seem quite right, but for every FADH2, you get 2 ATP, which seems weird because FADH2 has 2 hydrogen atoms, but NADH only has 1. It's because FADH is less efficient at giving up its hydrogens to the electron transport system. So keeping in mind that we can get 3 ATP for every 1 NADH and 2 ATP for every 1 FADH2, go back in your notes and find that table where we added up all of our goodies last time. Let's come back up here. I'm just going to scroll. Oh, I love those colors there. Here we go. I'm just going to scroll back here. Here we go. Okay, so this is where we added up all the ATP, NADH, and FADH2 from glycolysis, from the transition or intermediate reaction, and from the citric acid cycle. We found that we got 4 ATP total, which is diddly squat. Let me erase all this stuff. Here. There we go. Clean this up a little bit. All right, so if we get 3 ATP per NADH, so that's times 3, that's 30 ATP. And for every FADH, we get 2, so we're going to be multiplying that by 2. So 4 ATP from those 2 NADHs. If we add all this up, we have 4 plus 30 plus 4. That is a total of 38 ATP. Cool, huh? Now, in your homework, I talk about prokaryotes versus eukaryotes. There's a little bit of a difference here. If we take a look at the mitochondria here, so this is obviously a eukaryotic cell we're talking about here. It takes 2 pyruvates to get everybody in. It takes 2 ATP to get those pyruvates inside the mitochondria. So it's really going to be 36 ATP for eukaryotic cells and 38 ATP. I got to write over here because that's where there's space. 38 ATP for prokaryotic cells because they don't have a mitochondria to cross anything into. So we say generally 36 to 38 ATP. Yeah. So there we go. So, whoa, back when I said, wow, the cell went through all this crap to get 4 measly ATP, and what the heck is it doing with all these 10 ADHs and FADH2s? Wow, we get way more ATP. 30 plus 4, 34 ATP from these 10 NADHs and 2 FADH2s. Way more than just this measly 4. Yeah, because it's all about this electron transport system setting up that concentration gradient with the protons that releases energy when the protons equalize the gradient through ATP synthase. ATP synthase physically turns, basically, and it uses that energy to put the phosphate group back onto ADP to make ATP. Best bedtime story ever. So, but we're not done. Why aren't we done? We'll take a look at our equation here, our chemical reaction. C6H12O6 plus 6O2 yields 6 CO2 and 6 water. I have not seen anything with water yet. Have you? I mean, with O2? Has O2 gotten involved yet? Or did we produce a bunch of water yet? No, not at all. This has not happened. This happens way last. Happens way last. After that whole concentration gradient diffusion chemiosmosis thing. Way after chemiosmosis. Way after. So, oh my God, we're not done. Let's take a look at what oxygen actually does. So let's go back here I'm going to erase a whole bunch of stuff to give us some more room here. But let's say, okay, these protons already set up the gradient, and ATP was made, you know, when the protons released energy by diffusing back through, and everyone's like, yay, and everyone's happy, and we made a bunch of ATP. Big whoop-de-doo. Well, let's say that, okay, we started off with 10 protons up here, 10 up here, and 0 down here, so we need 5 to come through and equalize the gradient. Let's do that. 2, 3, 4, 5. There we go. 1, 2, 3, 4, 5 up there, and let's say 5. 1, 2, 3, 4, 5. Okay, so now we're at 5 and 5, so we're good. Is there a concentration gradient anymore with 5 and 5? Nope. It's equal. So no more protons are going to flow on through here. Right? Yeah, that makes sense to me. It's equal, it's equal. It's at equilibrium. No protons are going to flow through. And ATP synthase is like, but I need you to flow through so that, why did I do that? I had it on eraser. And ATP synthase is like, but we need more ATP, and for me to make more ATP, I got to turn some more. And I can't turn some more unless these protons come on through. And they're like, well, we're stuck because we're equal. And the cell's like, crap. Now what do? We need ATP, but we can't get any more of these protons to flow because we've got an equal number of protons inside as outside. What do we do? And the cell's like, you know, wouldn't it be cool if we could just erase these the way Mary just erased them there? If we could remove the protons from inside the mitochondria like that, now don't we have 5 up here and 0 down here? And yeah, so a few more of these protons would flow on through. Well, the cell can't quite just make them disappear, but it can make those protons change form. Let me put them back there. The cell's like, we could make all these protons disappear if we bind them to another molecule. Who are we going to bind these to? Oxygen says, I got this. I'm here. Actually, this oxygen just says blue to me. I'm going to change it to blue. Oxygen's like, ooh, pick me. I'll bind to the protons. But chemistry says, well, it doesn't work that way. Where a proton goes, electrons got to go with it. And meanwhile, where are some protons hanging around? Way back here. These protons are like, well, where the heck have you been? I'm sorry, these electrons here, these are electrons. The electrons are like, hey, proton, where have you been? And the proton's like, oh, I did this great little chemiosmosis diffusion dance thing here and made a whole bunch of ATP for the cell. What have you been up to? And they're like, well, I've just been bounced around from protein to protein over here, playing hot potato, waiting for you to get back. And oxygen's like, I'll take you both on, okay? So, when you get electrons plus protons, that makes a hydrogen atom, right? You bind that hydrogen atom to oxygen, and what do you get when you get oxygen plus hydrogen? You get H2O. Okay, and to balance that, let's see, we need 4 of these electrons, 4 protons. Yeah, I'm 102, so I got to put a 2 there. So there we go. Think about it. So one molecule of O2 can take up 4 of these hydrogen ions. 1, 2, 3, 4. Now do we have a gradient again? Oh yes, we do. Now we have only one inside the mitochondria, and let's see, we got rid of one here. Oh, and now we have, no, we didn't. Now we have 5 up here and 1 down here. That, friends, is a concentration gradient all over again. So now a few more of these protons can flow through. ATP synthase will turn, and it will use that energy to make more ATP. So what does oxygen do? We actually say that oxygen keeps the gradient going by removing protons from the mitochondria so that more of them can flow through. Oxygen maintains the gradient. We actually say, like, if you go to Molecular Oxygen's wiki page, believe me, it has a wiki page, it will say something like, oxygen is the final electron acceptor in aerobic cellular respiration. O2 is the final electron acceptor in aerobic cellular respiration. It does something. I mean, all of us know that oxygen is something that our cells need, but what does it do? Well, think about what happens if oxygen's not there. If there were no oxygen to bind to those 4 electrons and protons, the protons would flow through ATP synthase. We'd have 5 in here and 5 out there. It would equalize, and then we wouldn't get any more ATP because ATP synthase wouldn't have the energy from it anymore. We need oxygen to make ATP for our cells. If it's not there, that gradient gets equalized and we are up a creek without a paddle. No more oxygen, no more ATP. This whole thing gets gummed up. In fact, let me erase all this stuff over here. If the gradient equalizes and there's no oxygen there, NADH and FADH2 are like, hey, come on, electron transport system, we want to give you some hydrogen atoms in the form of electrons and protons. And this protein right here is saying, I can't, because it gums up the work. No more water, no more protons are flowing over the dam anymore. So if there's no oxygen around, NADH and FADH2 can't even give up any more hydrogen atoms to the electron transport system. They are screwed. So your cells need oxygen in order to make energy. Let's finish up our paragraph again, because I'm really liking this. And let's review it one more time. I hope stay with me. Don't check Instagram or your email on some other screen. Let's follow through here. During glycolysis, NAD plus and FAD remove hydrogen atoms from glucose, C6H12O6. They become NADH and FADH2, and they bring those hydrogen atoms to the electron transport system in the mitochondria. Once they're in the mitochondria, NADH and FADH2 give up the hydrogen atoms to the electron transport system in the form of electrons and protons. The electrons get passed from protein to protein while the protons get pumped across the inner membrane of the mitochondria. This is such an important sentence here. This creates a concentration gradient of protons that isn't stable. This is another important sentence here. Energy is released when the protons equalize the gradient through ATP synthase, and ATP synthase uses this energy to put the phosphate group back onto ADP to create ATP. These are, this here, man. This is so important. This is the gist of everything. This is the gist of chemiosmosis. All right, but while you're taking notes, and again, I really hope you're taking notes on this and not just listening to me and looking and thinking, oh, for the exam, I'll just go back and listen to the video. You can have a much easier time if you write this stuff down in your own notes and you can flip through instead of scrolling through me yakking at you. All right, so this process is called chemiosmosis. Back to blue. After chemiosmosis, molecular oxygen maintains the gradient. By binding to 4 electrons and 4 protons to become 2H2. Oops, I did not write this correctly. Let me. Maintains the gradient by binding to 2 electrons. 4 electrons. My bad here. Getting way ahead of myself. 4 electrons and 4 protons. Let's add oxygen in there. And this all makes 2H2O to make water. Oxygen is so important and our cells can't survive without it, because if oxygen isn't present to bind to the electrons and protons after chemiosmosis, NADH, no more protons can flow through ATP synthase and we won't get any more ATP. So if oxygen isn't present to bind to the protons and electrons after chemiosmosis, no more ATP can be made. New color. NADH and FADH2 can't give any more hydrogen atoms to the ETS. And your cell is basically screwed if it can't make any more ATP by chemiosmosis. But your cells sometimes don't get as much oxygen as they need for very brief periods of time. Like what's supposed to be happening this week was in Boston, was the Boston Marathon, right? On Patriots Day, and that is obviously not happening during the pandemic. But as I mentioned to you a while back, I have a friend who works mile 17. And muscle cramps are something that happen to a lot of people on mile 17, and here's why. Let's say you're running a marathon, and your heart is working really hard to pump all the blood to all of the cells in your body that needs it, because your cells need the oxygen that your blood is carrying, right? Your red blood cells are carrying oxygen to your cells through your blood. And if your heart can't pump the blood fast enough, if your cells can't get the oxygen that they need fast enough, you're going to have a situation like this back up here where there's no O2. And NADH is like, come on, can't we get more O2 here so that we can do this chemiosmosis thing? And if your heart isn't working, if your heart isn't as efficient at pumping oxygen to all the cells that you need, and let's face it, when you're running a marathon, your heart is going to have a hard time keeping up the pace there for the demand for ATP. But what your cells can do, very briefly, they can't get all the energy that they usually get out of chemiosmosis. But when there's no O2 around, NADH and FADH2 can donate those hydrogen atoms to an organic molecule instead. Donate to an organic molecule instead. Now let's come back down here and write this again, because something happens when this happens. If oxygen isn't present to bind to the protons and electrons after chemiosmosis, no more ATP can be made. NADH and FADH2 can't give up any more hydrogen atoms to the electron transport system, but they can give the hydrogen atoms to an organic molecule instead. And the definition of giving hydrogen atoms to an organic molecule instead, this is fermentation. Your cells can ferment if they're out of oxygen very briefly. And here's the deal with fermentation. You get a little ATP, like 2 or 3 total out of that. None of this, 30 from 10 NADHs and 4 more from FADH2s. You just get 2 or 3 total. You get a little bit of ATP from that. But it's kind of like an last-ditch effort for your cells if your heart can't keep up with the demand of running a marathon. But here's the issue. When human cells ferment, we produce lactic acid. When human cells ferment, we get 2 to 3 ATP, which, isn't much, but it can be enough just to let you, run a few more steps. We get 2 to 3 ATP and produce lactic acid, which we already talked about last time when we were talking about enzymes. That gives you muscle cramps. Ouch. And that's what my friend sees a lot of on mile 17 of the marathon. Right. Not every cell that ferments will produce lactic acid, though. Yeast cells, when yeast cells ferment, and they actually prefer to ferment all the time, they get a little bit of ATP, but they produce ethanol. Actually, and CO2, right? Probably know that from your lab reading. Right. So, fermentation in yeast produces ethanol, alcohol, which we like. This is how beer and wine are produced. Beer and wine are fermentation products. Cheeses are produced through fermentation. Yogurt is a fermentation product. Sauerkraut. We talked about sauerkraut when we were still having in-person classes. That's a fermentation product. Even chocolate, I didn't know this till a couple years ago. Chocolate is actually produced, cocoa beans need to lie out and ferment for a while, or the chocolate turns out to be very bitter. So chocolate is also a fermentation product. Not only yeast cells ferment, yeast cells and tons of bacterial cells will ferment as well. So bacteria such as lactobacillus, which is present in yogurt cultures, for example, they ferment as well. They just don't produce ethanol, but they do produce acids. Yeast is kind of difficult or commercial yeast, I should say, is difficult to come by. So a lot of people, including myself, are using sourdough starters, where you're just using wild yeast and microbes from the air to help raise your bread. The CO2 or the air bubbles, and that helps raise the dough, but the ethanol and also acids in the sourdough starter will help develop the gluten. So sourdough starter, it's basically wild yeast plus some bacteria, usually lactobacillus, because it can stand the acid. Yeah, so the fermentation process is really important to civilization. It's hugely important to the food industry, but your cells will do it as well if there's no oxygen around. So in fermentation, really, the definition of fermentation is when an organic molecule is the final electron, or let's say it's the final hydrogen acceptor. In aerobic cellular respiration, oxygen is the final electron acceptor. I had written it down and erased it. What the heck? Let's write it down here. In fermentation, an organic molecule, sometimes it's pyruvate, is the final electron or hydrogen acceptor. You get a little ATP from it. That's enough to keep a yeast cell or a bacterial cell going, but it's not enough to keep a human cell going. So in aerobic cellular respiration, O2 is the final electron or hydrogen acceptor. You get 36 to 38 ATP as opposed to 2 or 3 up here for fermentation. Okay, so aerobic cellular respiration, this is, aka chemiosmosis, right there. Chemiosmosis gives way more ATP, 36 to 38, than fermentation does. So your cells really, really need O2. Oh, gosh, you need it. But we can get by for a little bit. That's why people get muscle cramps. Your cells aren't getting the oxygen they need when they need it right away. Or your cells' ATP demand is higher than the amount of oxygen that can get into your cells. So that's my story. All right. Oh, wait. One more story for you. Why not? You know how at the beginning of school years, usually late September, after school started for a couple of weeks, in normal times, and hopefully we'll get back to that again, how public schools will have these things called open houses. And they're usually in the evenings, and family members can go and visit their students' teachers, and you get your student's schedule, and you follow your kid's schedule, and you go around and you go to math class, and the math teacher tells you, oh, this is what we're doing in class, and you go to the English class, and you sit in the little chairs that your students sit in, and you're like, oh, here's what we're doing in English class. And I went to my son's biology class I went to my son's high school open house one year, and this was, I think, his freshman or sophomore, 9th or 10th grade, I don't remember. But I went to the biology room, and the biology teacher's like, oh, here's what your students have been doing. We did a leaf study, and, we studied some erosion. We're looking at fossils, too, and we're going to be talking about cells throughout the year and everything. And as we talk about cells, we'll talk about how cells work and what they need. And I just want to put this out to you, the teacher said, to all you family members here, and ask you if why do our cells need oxygen? Why do we need to breathe oxygen anyway? What does it do? And I was like raising my hand going, pick me. It's that, well, oxygen is the final electron acceptor in aerobic cellular respiration. Except I didn't say that. Thank you, Hermione Granger. I didn't think I would move the conversation too well there. But another parent, and I knew this guy because he was the parent of one of my son's friends too, so I knew them well, and raised his hand and said, oh, oxygen has something to do with how our cells make energy. And I was like, hot damn, that's really good. This person was not a scientist, didn't study science in college, but he had really good science learning there, some really good teaching there. He remembered after all these years that it's oxygen that's responsible for binding to the protons and some electrons, and in the process forms water that keeps chemiosmosis going. That it keeps this gradient going so that more protons can flow through, turning ATP synthase some more so that it can make more ATP. Our cells need O2 to make energy. Otherwise, chemiosmosis doesn't happen. Sure, our cells can do this fermentation thing a little bit, but that gives us just a piddly little 2 or 3 ATP. We need more than that. We are multicellular organisms that need the 36 to 38 ATP that chemiosmosis gives us. And chemiosmosis doesn't happen without oxygen. Chemiosmosis requires oxygen to be that final electron hydrogen acceptor. And that is the best bedtime story ever. And that's enough. That's enough before our April 23rd exam. All right. So there we go. So the April 23rd exam is going to pick up where we left off from exam 2. So everywhere from our March 31st online lecture to today's online lecture. March 31st, this was our first online lecture. So basically everything on there, after I talked about viruses and why antibiotics don't work for viruses. So this started with transport across the membrane. And that was diffusion and concentration gradients and osmosis and hypertonic, hypotonic, isotonic stuff. We also talked about active transport and the sodium-potassium ion pump. And then we got into energy, how cells make energy, and we talked about the laws of energy, laws of thermodynamics, and how they apply to what we're talking about in the cell. We spent a ton of time talking about enzymes, how they work, what stops them from working, what denatures enzymes. We also talked about types of reactions, exer and endergonic. We also talked about oxidation and reduction reactions, and then the whole process of cellular respiration, glycolysis, intermediate step, citric acid cycle, and electron transport, and also what NAD plus and FAD do, and also, the whole process of chemiosmosis and what oxygen does. And then finally, fermentation. You even have a homework, and it's also due on Thursday. So the homework is due Thursday, April 23rd, and the exam is April 23rd. Online, make sure you upload your homework. I would just do it as a Word file. Just fill in the blanks. That way you won't have to print it out. If you want to print it out and turn it into a PDF and upload a PDF, that's fine too. I did not, although, you know, there are size limits. So if you take a picture of something and just want to upload the photo, you got to make sure that it is not super high res or a super big file, because Moodle isn't going to take, there's a file limitation there, and I'm not sure what it is. All right, so you need to upload your homework. Take the exam on the 23rd. I am opening up at midnight on the 23rd. It is closing at 11:59 p.m., so that's 2,300 hours and 59 minutes that I'm closing it. Got to take it that day. It is open note. Please make sure that you have taken notes. You are not going to want to have to Google your way through this exam. I mean, yeah, don't do that. Anyhow, and so let's see, you've got this homework due and you're doing it on Thursday, and then we'll start on some new stuff the week after that. So this is plenty for the day, and if you have any questions, please post them in the comments, and I will be answering questions through, let's say Wednesday afternoon, Wednesday the 22nd. I hope you are well. I hope you are safe, and take it easy. Keep on swimming. Just keep swimming. Just keep swimming. And I'll see you all. Bye-bye.